Hi and hello everyone. Just today we are here with one of the most important topic of microbiology, and we are going to discuss and talk about the difference between gram positive and gram negative bacteria. So now coming to the history of all this, there was a scientist from Denmark and the name was H.C. Gram, and he was working in Berlin. So in 1884, that was a historical moment, that was a remarkable research or discovery in the field of microbiology. In 1884, the gram staining was developed by H.C. Gram, and that step was just a milestone and remarkable research work in the field of microbiology. And on the basis of this gram staining, the world was now in condition, the world was capable to differentiate, to classify in a standard technique like a bacterial population in a gram positive category or a gram negative category. So in a detail we are going to discuss about like what is the difference between gram positive bacterial cells and gram negative bacterial cells. So starting with gram positive bacterial cell. So this is the cross part of the cell, like cell membrane, cell wall part of the bacterial cell. So one by one we are going to check like what are the structure and what are the components that are present in this bacterial cell. So coming from gram positive bacteria, the main part is peptidoglycan. You can see this is the peptidoglycan, and this peptidoglycan, it is made of two main components. That one component is NAM and another component is NAR. So N-acetylmuramic acid and N-acetyl glucosamine. So both these components just join with peptide, four of the peptide layers, like four amino acids together make a peptide chain, and that makes integration of all this peptidoglycan layer. So this peptidoglycan layer, it is like a thick layer in gram positive bacteria, and after the peptidoglycan there is inner cell membrane layer, and it is made up of phospholipid just adjacent to cytoplasm. So phospholipid, then a thick layer of peptidoglycan, which is about 80 to 90% part of the gram positive bacteria, and now embedded in this peptidoglycan layer is teichoic acid and lipoteichoic acid. Now coming to teichoic acid and lipoteichoic acid, it plays a major role in cell division of the gram positive bacteria, as well as providing resistance to the extreme conditions, like resistance to extreme salt conditions or resistance to extreme temperature conditions. So it is very helpful for gram positive bacteria. So this is overall the structure of the cell wall of the gram positive bacteria. So you can see this. Now coming to gram negative bacteria. So like it's a little bit complex compared to the gram positive bacteria. So now coming from upper to lower part. So the upper part, it is made up of that is lipopolysaccharide, and in between lipopolysaccharide there is porins for transfer of some of the materials or components that is required for metabolic activity of gram negative bacteria, and some embedded proteins are also there. So after this outer membrane made up of lipopolysaccharide, there is a thin layer of peptidoglycan. You can see it is a thin layer of peptidoglycan in the gram negative bacteria. Now comparing it with the gram positive, there is thick layer of peptidoglycan. It is easily visible here. Again, NAG and NAM units are involved in the making of peptidoglycan layer here. And now the innermost layer it is like cell membrane. It is made up of phospholipid again, similar to the gram positive bacteria. But there is periplasmic space, this one, between outer and inner cell membrane. There is periplasmic space consisting of like thin peptidoglycan. So you can say it is like triple layer compared to the double layer of gram positive bacteria. So like there are several differences, and one of the most important part of this gram negative cell wall is like lipopolysaccharide, and it is just composed of three components like O antigen, then O polysaccharide, and then the inner part is lipid A. So in this one, O antigen, when it comes in contact of the host, like human being, it triggers the immune system inside the host. Coming to the lipid A part is like one of the most important part that plays act as a endotoxin when goes inside the human blood. So endotoxin shock it causes, and that's the reason that after fragmentation of gram-negative bacteria in several sources, when this lipid A part goes in the bloodstream of human being, it acts and works as a endotoxin, and it produces endotoxin shocks inside the body. So now coming to the main differences. So what are the main differences when we compare this one? So in gram-positive bacteria, peptidoglycan layer is thick. It's almost 90% part of the cell wall, against the gram-negative where the peptidoglycan is very thin and only 5 to 10% part of the cell wall. Now coming to teichoic acid and lipoteichoic acid, within the peptidoglycan layer, it is present in only gram-positive bacteria. But in gram-negative, teichoic acid and lipoteichoic acid are just absent. Now coming to lipopolysaccharide, it is absent in gram-positive, but lipopolysaccharide as a part of lipid A, main component of it, that act as endotoxin as part of gram-negative bacteria. Now coming to exoenzymes for metabolic activity and dissolution of large components that is required for gram-positive bacterial cell metabolic activity. Exotoxins are released and that dissolve the matters, and it slowly passes through the cell wall inside the bacterial cell in gram-positive bacteria. Now coming to gram-negative bacteria, they use periplasmic enzymes. So these enzymes support in passage of some of the components, and these enzymes that break down and dissolve in the simpler form from the periplasmic space. Now coming to gram staining. So based on gram staining, gram-positive bacteria appear as a purple colored cells in the microscope, against the gram-negative bacteria that appears as a pink colored cells in the microscope. So this is all about the gram-positive and gram-negative cell wall and basic staining color difference. So further we will just check the methods of gram staining and how it works, how it stains the bacterial cell in the next episode. So okay, thank you for watching it and hope it will be helpful for your knowledge. Thank you.